3D printed porous titanium for smart implant. Presented by Senior Design Group 24. Alexis, Mateo, Sarah, Allison, and Sharon.
There are a few reasons why bone implants are essential for the medical world. To start, there's a link between aging and increased risk of bone diseases and bone weakness. Some common issues with current bone implants are that the implant is not as elastic as bone, does not integrate well with the bone, which causes further issues like stress shielding and implant loosening. Our solution to these current issues involves creating a solid core in the interior, which will be surrounded by a porous titanium framework. The interior of the implant has gelatin-based hydrogel, which will be graded for proper osseointegration. The design also contains microfluidic channels and a 3D printed porous structure, which allows for nutrient and waste transportation.
Our group developed a porous structure based on the Gibson-Ashby open cell foam model. This was because of the low elastic modules of the bulk printed EBM. As we can see in the table, the metal foam would have been able to meet the elastic modulus range found in human femur bones. The table shows the implant would prevent stress shielding in both an ideal case and in applications where the EBM would introduce processing defects into the structure and lowers its overall C1 value by meeting that of human bone. The image in the top center shows a SolidWorks file we were going to implement in printing. The design lowers both weight and cost and meets all the design specifications.
Our choice of hydrogel is bovine gelatin type B. The advantage of using this is that it's inexpensive, and also gelatin mimics the body's ECM. It's also biocompatible, biodegradable, and useful for cell seeding. The disadvantage of gelatin is that it's not very strong. Due to this, physical cross-linking and UV cross-linking needs to occur.
To make the gelatin hydrogel, a procedure was developed based off of a few procedures found through research. It is a two-part process, and you can see here the first part. First, gelatin and methacrylic anhydride are mixed with PBS while heated. Then the solution reacts for 1 hour in the dark while still being heated to allow the gelatin and methacrylic anhydride to bond. Then the solution is poured into containers and prepared to be freeze-dried. Liquid nitrogen is used to freeze the solution, and it takes a few minutes for it to completely freeze. The vials are then ready to be placed in the freeze dryer. Once loaded in a large beaker and attached to the machine, they are left to dry for 48 hours. After the 2 days drying, the freeze-dried powder is removed from the machine. The powder is then dissolved completely in DI water while being heated. After it has completely dissolved, photoinitiator is added, and then it is immediately poured into a mold and exposed to UV light for 30 minutes. Finally, the solution is left to react at 4 degrees Celsius for 24 hours to fully cross-link. Here you can see the development of the hydrogel as the procedure was improved. There is a noticeable increase in transparency as well as reduction of small white undissolved particles. The most recent hydrogel is the most transparent and was solidified so it could be handled. Some changes that were made to the procedure were increasing stirring times and increasing the temperature used to dissolve the freeze-dried powder.
The importance of the migration assay is to study how cells interact and migrate within the hydrogel and eventually within the femur implant, since ensuring osseointegration is one of our main focuses for this project. Included here is our migration fixture. It's composed of a few sheets of acrylic that will sandwich our hydrogel, and the cells will be seated directly on top of it. It appears brown here, but that's just the protective coating. The acrylic itself is transparent and will allow for us to monitor and view the cells moving underneath a microscope. In our migration assay, a high concentration of FBS will be inserted into the bottom of the fixture, separated from the lower concentration of FBS by the hydrogel. Cells will move through the hydrogel towards the high concentration of FBS, and their movement can be tracked and quantified using the live imaging microscope.
Ideally, we would have used human osteoblasts as they would be best at assessing osteointegration. We decided to use human dermal fibroblasts because we already had access to them. Also, fibroblast behavior is similar enough to osteoblasts that we are confident in their accuracy for testing purposes. The video on the left shows the movement and division of fibroblasts cultured in a petri dish. The video on the right shows fibroblasts cultured on our gelatin hydrogel. For future work, we would like to perform the assay as originally planned, incorporate the hydrogel into the titanium structure, perform a viability assay on titanium as a proof of concept, and 3D print an implant model out of PLA. In conclusion, we developed a titanium scaffold with optimal mechanical properties for our application. We developed a procedure for fabricating a gelatin hydrogel that is ideal for cell growth. We designed a cell migration assay for studying theoretical osseointegration, and we live monitored cell behavior on the gelatin hydrogel.
We learned basic procedures for both monolayer cell culture and cell-seeded hydrogel culture methods. We developed procedures that include a freeze dryer to create hydrogels and a live monitoring microscope to record images of cell migration on said hydrogels. Throughout the project, our group learned material synthesis techniques to better develop the hydrogel and modeling techniques, such as finite elemental analysis, to propose a mechanically stable metal foam scaffold.